Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we were discussing about different cellular signaling pathways in which we have covered some pathways like RAS MAP kinase pathway, the TGF beta signaling pathway, Notch delta signaling. Today we are going to discuss about Wnt signaling pathway. So this is a very unique pathway in itself because this pathway also works through latent transcription regulator, but the latent transcription regulator, which mediates this pathway, has some other roles as well in the cell for cell adhesion. So today we are going to learn about Wnt signaling, which is an extracellular signal protein, how it is very unique because it has hydrophobic fatty acid chain in it, and how with the help of beta-catenin it is going to mediate the transcription of Wnt responsive genes. Okay, so the today's topic of discussion is Wnt signaling. So this protein Wnt was identified in both Drosophila as well as in mice. So in the Drosophila, a gene named wingless, okay, this gene encodes the protein Wnt, while in the mice, a gene named INT1 encodes this Wnt protein, and that is why this protein is also written as WNT, okay, and is pronounced as Wnt. Okay, so there are almost 19 types of Wnt proteins found in humans, which have very distinct functions, but those functions are overlapping in the cell in the biological systems. So this Wnt protein activates a protein known as beta-catenin, which is a intracellular signal, and beta-catenin will do the downstream job, and it is going to activate Wnt responsive genes. Okay, so let's learn about this pathway in detail. So this Wnt protein is, I told you that Wnt protein is very unique because this protein is encoded by a gene, and after the this protein is formed, when it is being processed in endoplasmic reticulum, a fatty acid chain is attached to this, it is covalently attached to this protein, and this fatty acid chain gives it a hydrophobic nature. So this is fatty acid chain, and due to this fatty acid chain, this Wnt molecule has a hydrophobic part, and that is why this it makes it difficult to get dissolved in the extracellular matrix and to traverse through the extracellular matrix freely, and that is why this Wnt protein usually is found in the cell membranes because it has a hydrophobic part as well. So this fatty acid chain, it is attached to this Wnt protein in ER. So this is Wnt molecule, which works like an extracellular signal. So when it works as an extracellular signal, it is going to activate beta-catenin downstream. Okay, so there are two scenarios when Wnt is absent. When Wnt is absent, this beta-catenin is, so beta-catenin is always present in the cell, but if the Wnt signaling is not there, the beta-catenin which is formed, which is being transcribed and translated, this beta-catenin will be immediately degraded by a proteasomal degradation complex. Okay, if the Wnt signaling is not there. So what does Wnt signaling do? When Wnt comes and it signals the receptor molecules present on the cell, it inhibits the proteasomal degradation of beta-catenin so that the beta-catenin can go and it can translocate from the cytoplasm to the nucleus, and it can do its own downstream job. Okay, so there arises two scenarios. One where when the Wnt signaling is absent, means the Wnt protein is not present in the extracellular matrix, in the extracellular environment of the cell. So Wnt is absent. And the other scenario is that Wnt is present. So when Wnt signaling molecules, they are absent in the cell, they are not activating receptors. So what happens is that the beta-catenin, which is present inside the cytoplasm, will get degraded. So beta-catenin gets degraded, it gets degraded with the help of a proteasomal degradation complex. Okay, so this proteasomal degradation complex has mainly four different type of proteins. The first one is CK1, which is a casein kinase. So this casein kinase is a serine kinase. So so first job is done by CK1. It phosphorylates beta-catenin on the serine residues. So it is a serine kinase. Okay, so it phosphorylates the beta-catenin over the serine residue. Then comes other protein known as GSK3, which is a glycogen synthase kinase 3. So this GSK3, first the beta-catenin will be phosphorylated by CK1, and GSK3 will also phosphorylate beta-catenin on some other residues, and the phosphorylation by both CK1 and GSK3 will now tag this beta-catenin for ubiquitination. So due to the phosphorylation, so first event is phosphorylation by CK1, then the phosphorylation by GSK3, and due to the phosphorylation of beta-catenin, now this beta-catenin will be tagged for ubiquitination. So ubiquitin ligase will come and it will add the ubiquitin group over the beta-catenin. So now once this beta-catenin is it is marked by the ubiquitin, means it is meant to be degraded. So a proteasomal complex will degrade it, and that in that complex two proteins known as axin and APC, they are going to act as the scaffold protein, so that they will provide a platform for this whole complex to be able to adhere and to be able to get attached to those proteins. And that is why axin and APC is adenomatous polyposis coli, adenomatous polyposis coli. So this is different from APC, which has a role in cell cycle. So though both of them, they are the scaffold proteins over which the those beta-catenin, CK1 and GSK3 as well as ubiquitin ligase, those proteins are going to adhere themselves. So those are axin and APC are scaffold proteins, right? So this particular proteasomal complex is going to degrade the beta catenin. So beta catenin will be degraded, and now if there's no beta catenin, the Wnt signaling will not go on. So generally the Wnt responsive genes that are present in the inside the nucleus, so Wnt responsive genes. Okay, so in when there is no Wnt signaling, those responsive genes are silent, right? So they are called Wnt responsive genes because they get activated in response to Wnt protein, Wnt extracellular signal. So whenever Wnt is present there, those Wnt responsive genes, they are going to get transcribed. But we are discussing the first scenario when Wnt is absent. This means the Wnt responsive genes, they are silent. So for keeping them silent, certain inhibitory complex is working, right? So that is a given fact that some inhibitors are going to work over the Wnt responsive genes. So those inhibitors which work on them are LEF1 and TCF complex. Okay, so these are the inhibitors or the repressor which are repressing the Wnt responsive gene and which need a co-repressor which belongs to Gaucher family. So Gaucher family proteins, they act as the co-repressors. So this whole complex of Gaucher family and LEF1 TCF complex, this whole complex is going to inhibit the Wnt responsive genes whenever Wnt is not there in the biological system, in the cells. So when the Wnt acts as the extracellular molecule, this complex will be broken down with the help of beta-catenin. We'll see how it is done. And due to the inhibition of this inhibitory complex, the beta-catenin, it removes the Gaucher family and it works as a co-activator. Okay, we'll discuss that in a while. So Wnt responsive genes are silent due to this particular inhibitory complex in the absence of Wnt. So what happens when the Wnt is present in the cell? So when Wnt is present in the cell, that will be sensed by some receptors, right? So this signaling is again unique in its way because generally we talked about receptor proteins which receive the extracellular signal molecules. They usually form dimers or they form tetramers, or the certain part of them, the cytoplasmic tail, is going to be cleaved and which is going to act as an intracellular signal. Or somehow, with the help of molecular switches, the receptor proteins, they are going to form some intracellular signaling molecules, or they are going to activate intracellular signaling molecules to carry out the intracellular signaling cascade. But Wnt protein is unique because the receptor proteins that are present on the cell for the Wnt protein, there exist two receptors, and they together are receiving Wnt protein, and they are forming a co-receptor complex which will be activated to do the downstream signaling. So this is a very unique part. So in the plasma membrane, there exist two proteins. So this is the plasma membrane, and on the plasma membrane, the two proteins exist. Okay, so there's a single pass transmembrane protein. Okay, so that protein is known as LRP, which is the main protein for the Wnt extracellular molecule, and there exists other 7' transmembrane protein. Okay, well, okay, so this 7' transmembrane protein, it looks something like this. Okay, the figures I draw, they are very important, so that because they are going to represent the whole schematic in a way. So this is a 7' transmembrane protein which is known as frizzled, and it resembles a lot with the GPCRs, right? So G protein coupled receptors, they were also 7' transmembrane proteins. In the same way, frizzled receptors are also the 7' transmembrane protein. So both of them are crucial. The LRP as well as frizzled receptor are crucial so that they can receive the Wnt extracellular signal. So this Wnt protein, as discussed before, okay, so this is Wnt protein, and it is being coupled with the fatty acid chain. So fatty acid chain is covalently attached to this Wnt protein. So this fatty acid chain, it gets received by frizzled receptor, while the this Wnt part or the other domain reacts with the LRP receptor. Okay, so when Wnt signal comes, the fatty acid chain is going to react with frizzled receptor, and so let me just draw this LRP when it receives the Wnt. So this is LRP, and this is Wnt protein. So here you can see the particular domain of the Wnt protein it is reacting with the LRP receptor. LRP receptor is LDL related protein. LDL is low density lipoprotein, right? There exists LDL, HDL. So this is LDL related protein which acts as a receptor molecule for Wnt along with the frizzled receptor. So this frizzled receptor and LRP, both of them get activated when the Wnt is present in the cell. So both of them gets activated, and the activation of both of them forms a dimer. But here in this case, two different receptor molecules are forming dimer, and that is why we will call it as co-receptor dimer. Okay, because here the LRP is the main receptor, while frizzled is acting as the co-receptor of the Wnt molecule, right? So once the Wnt attaches to this frizzled as well as LRP, both of them, it will activate the CK1 and GSK. So LRP, so this will the co-receptor dimer complex formation. So co-receptor dimer formation will activate CK1 and GSK3. So those activated CK1 and GSK3, both of them are kinases, so they are going to phosphorylate LRP. So now LRP will get phosphorylated. So here I'm adding phosphate residues over the LRP. So the LRP is getting phosphorylated due to the CK1 and GSK3. Okay, so the activated, how does CK1 and GSK3 gets activated? They gets activated due to this co-receptor dimer formation. So they will phosphorylate the LRP receptor, that is going to further activate a protein known as disheveled. Okay, there exists a protein known as disheveled. This disheveled protein will get activated, and it will attach itself to over the frizzled receptors. Okay, so this activated disheveled receptor is very important because it works as a scaffold protein, okay, for multiple intracellular signaling proteins. And in this case, disheveled protein, it recruits axine protein, and it inactivates the axine protein. So what will happen? CK1, GSK3, they are going to phosphorylate LRP, and phosphorylation of LRP will result into the activation of disheveled protein, and disheveled protein will get recruited to the frizzled protein. Okay, so this get recruited to the frizzled protein, and this recruitment will activate disheveled protein, and the activation of disheveled protein will recruit axine protein. Okay, so axine protein complex will be recruited over the disheveled protein, and it will get inhibited because now it is the axine protein complex which was going to get bind to the beta catenin and serving as the scaffold protein. So that was the function of the axine in the absence of the Wnt. But due to the Wnt, the disheveled protein it is getting activated, and it is going to recruit the axine protein complex. So the axine protein complex is already engaged with disheveled, and that is why it is not free to work on beta catenin, and that beta catenin is now it is stable inside the cytoplasm. It won't be degraded because basically the actin protein complex, which was holding all those CSK3 and so CSK1 and GSK3, it is not there. It is already engaged with some other protein called as disheveled, and that is why the beta catenin will be stable. It will not be degraded in cytoplasm, and that is why this stable beta catenin will now migrate to the nucleus, and it is going to do its downstream job. So this is how the Wnt signaling activates a protein disheveled, and it results into stability of beta catenin inside the cytoplasm. Okay, so beta catenin, which is now stable, it is going to go, so it is going to migrate, or it is going to translocate into the nucleus, right? So in nucleus, what will it do? It will go and bind to the LEF and TCF complex, LEF1 TCF complex. This will bind with beta catenin, and means it is going to replace the Gaucher protein. Okay, so this replacing of Gaucher protein will result into beta catenin working as a co-activator. So before LEF1 TCF was forming a complex with Gaucher, and it was working as an inhibitor because Gaucher was a co-repressor. But now beta catenin will work as a co-activator with this LEF1 and TCF, and this whole complex will be now activator complex, and this activator complex is going to activate the Wnt responsive genes that are going to transcribe the Wnt target genes. So they are going to transcribe the downstream genes present on the DNA. So this is how beta catenin works as a co-activator for the Wnt responsive genes. So this is the whole signaling pathway. So I told you that beta catenin is a latent transcription regulator, means it is going to migrate to the nucleus when it is stable inside the cytoplasm, means it is not degraded. So it will migrate to the nucleus, and it is going to activate the transcription. So here you can see, due to the Wnt signaling, the transcription repression. So Wnt responsive gene were first repressed, they were silent. So transcription repression is converted into transcription activation due to the presence of Wnt signaling. So the beta catenin molecule is a very distinct molecule inside the cell because it just not take part in the signaling pathway and activate certain genes, but it is also important in cell-cell adhesion. So this is beta. If the question arises that where is beta catenin found inside the cell, so it is found at two main sites. First, in cytoplasm, if the Wnt signaling is there, it will get activated, and if Wnt signaling is absent, the cytoplasmic beta-catenin will be degraded. Simple. And the other site where the beta-catenin is present and it is active is over the cell-cell adhesion site. So it is going to mediate cell-cell adhesion. So it is going to present over the cell-cell junctions, and it is going to mediate the cell-cell adhesion. So beta-catenin is seen in both Wnt signaling as well as for the adhesion of the cell to the other cell, right? So that is very important. So this is the Wnt canonical pathway that I discussed here. So let's discuss this whole pathway by the help of schematic. So this schematic was given, it is given in the Bruce Albert, and it beautifully explains the whole scenario. So here you can clearly see that when without Wnt signal, LRP and frizzled, they are inactivated, and due to the inactivation of both of those receptors, disheveled is also inactivated. So inside the cytoplasm, what is happening? The beta-catenin, it will get phosphorylated by CK1 and GSK3, because both of those proteins, they are, they are attaching, or they are recruiting themselves to the axin and APC molecules. Both of them, axin and APC, both of those proteins, they act as a scaffold protein, and recruitment of GSK3 and CK1 to those scaffold protein is going to activate those kinase domains of those proteins, and that is going to phosphorylate beta-catenin. So phosphorylation of beta-catenin will result into ubiquitination of this beta-catenin, and it will be degraded by the proteasomal complex. So this particular complex will result into degradation of beta-catenin. So now beta-catenin is degraded, the whole repressive elements that are present over the DNA, that will stay as it is. So Gaucher family proteins, they are acting as co-repressor. So LEF1 and TCF, it is binding, it is a DNA binding complex. So this complex is bound to the DNA, to the Wnt responsive genes over the Gaucher, which is a co-repressor, it is inhibiting this whole gene. So this whole element, so it is inhibiting, or it is silencing the Wnt responsive gene. So this happens when the Wnt is absent in the biological system, and when the cell signals for the Wnt, so means a certain cell, it is secreting Wnt proteins. So this Wnt protein here, you can clearly see, has two different domains, and they are binding to two different receptors. So this particular domain activates the LRP. So it is going to bind LRP and activate the LRP, while the hydrophobic fatty acid chain will be bound to the frizzled receptors, so that the frizzled receptors will get activated. So the Wnt binding will activate both LRP and frizzle receptor, and that is going to form a co-receptor dimer. So this formation of co-receptor dimer will activate both of the receptors, and that is why the CK1 and GSK3, both of the kinases, they are going to phosphorylate this activated LRP, and activation of this LRP will result into the activation of disheveled protein, and disheveled protein will recruit itself over this activated receptor dimer complex. So this recruitment of disheveled protein will result into attachment of axin protein to the disheveled protein. So now, as axin is preoccupied with disheveled, it cannot work over the beta-catenin, and that is why beta-catenin, which is present inside the cytoplasm, it won't be degraded, and it will migrate to the nucleus. So this is nucleus. So it is now migrated to the nucleus, and it is going to replace the Gaucher protein, the proteins which belong to Gaucher family, and due to the replacement of this repressor protein, the beta-catenin is going to now work as a co-activator, and it is going to activate the transcription of the Wnt target genes. So LEF1 and TCF plus beta-catenin, this whole complex will work as an activator complex to activate the Wnt responsive genes, and this is going to result into transcription of Wnt target genes. So one of the target genes of the Wnt signaling is MYC gene. Okay, MYC gene, which if is expressed, it is overexpressed inside the cell, will result into cancer progression, cancer formation. So tumor will be formed if the MYC gene is overexpressed inside the cell. We have already discussed in this in cancer biology. So one of the downstream genes for the Wnt responsive, so the one of the Wnt responsive genes is MYC gene, which will result into formation of c-MYC. and that is going to result into tumor formation. So by due to some factor or due to some reason, if inside the cell either the Wnt signals are high, means Wnt is overexpressed, or due to some reason, let's say that the cytoplasmic beta catenin is increasing, or it is not getting degraded even if the Wnt is not there. So let's assume that due to some genetic mutation, Wnt whenever the Wnt signal is not present, beta catenin is meant to be degraded, right? So let's say that there exists some mutation in the axine protein, or a mutation in in the disheveled protein. So the mutation in axine protein will result into, the mutation is such that the axine protein is not now, it is not working as a scaffold. This will result into inhibition of degradation of beta catenin, right? So if the axine cannot work as a scaffold protein, even in the presence of the even in the absence of Wnt signal, beta catenin will no will be stable, right? So the stability of beta catenin, I'm sorry, seen, this protein gets mutated and it does not act as the scaffold protein. So this will result into the stability of beta catenin. So beta catenin won't be degraded, and that will result into the activation of Wnt responsive gene even if the Wnt signal is not there. So that is going to result into formation of the Wnt of the cellular products which get formed due to the Wnt presence, as well as the production of c-Myc, and that will result into the uncontrollable cell proliferation leading to the tumor progression. So either the beta catenin is stable inside the cytoplasm whenever the Wnt signaling is not there. So what are the multiple reasons for the cancer progression? Either the higher expression of Wnt proteins, due to some reason Wnt is overexpressed inside the cells. So basically Wnt signaling is there, and that is why beta catenin won't be degraded, will result into transcription of Wnt target gene. The second reason could be that Wnt signal is not there, but due to some reason beta catenin is stable. So that the reason that beta catenin is stable could be either axine is mutated so that it does not work as the scaffold protein, or the disheveled protein is mutated such that it is active even if the Wnt signal is not there. Okay, so in both of the cases, the beta catenin won't be degraded if the disheveled protein is active, or the axin is mutated and it is non-functional now. So in both of the cases, the beta catenin inside the cell will be stable, and that is going to result into the tumor progression. So either Wnt is overexpressed, or beta catenin is overexpressed, or the axin is mutated, means it is non-functional, or disheveled protein, it stays active even in the absence of Wnt signaling. Either of those factors, they are going to result into the transcription of Wnt responsive gene unnecessarily, which is going to produce c-Myc, resulting into uncontrollable cell proliferation leading to the cancer. So this is how Wnt signaling is very important, and it somehow works like notch signaling, because it is before the notch signaling the genes were off and now they got switched on. In the same way, Wnt signaling also, due to presence of Wnt, the transcription repression gets converted into transcription activation. So I conclude the lecture here. In next lecture, we will learn about hedgehog signaling. So we have still we have a lot of pathways to learn, such as hedgehog pathway. So we'll learn that in next lecture. Till then, see you. Bye bye.